Okay, so if everyone can hear me okay, we're good? Okay, so hi everyone, I'm Lara. I'm a fresh postdoc as of January of this year at Ghent University, where I also received my PhD in December, and I'll be talking to you today about some of the work that came out of that, namely community structure of deep sea nematodes in the Clarion Clipperton Fracture Zone using a metabarcoding approach. So next slide, hold on, I think my slide isn't working, just a sec. I'm not able to advance to the next slide, but working with sequencing data means that you typically don't get to see the actual thing, so I wanted to add some pictures of our protagonists. So nematodes were named as such due to their likeness to a thread, which is the word nema in Greek. Nonetheless, the phylum has diversified beyond the thread look into a variety of body plans, such as the caterpillar or the periscope. We know that today 80% of multicellular animals are nematodes. They are extremely abundant and ubiquitous and can be found from the polar environments to the tropics to deserts, freshwater lakes, and the deepest of ocean trenches. And one particular representative of the phylum, Meiobenthologists elegans, is of great importance to both the molecular and neuroscience world as a model organism, and is the only animal for which we have both described the genome and the connectome, which is a map of all neuronal connections in Meiobenthologists' brain. Today we'll be talking about free-living marine nematodes in the Clarion Clipperton Fracture Zone, which I will call the CCFC from this point on, and that is situated toward the west of Mexico. So this is our study area. The CCFC looks like this. It's a vast abyssal plain, a sedimentary environment littered with these stone-like formations, which are polymetallic nodules. They form over millions of years through the precipitation of metals onto a central nucleus in a similar way to how pearls are formed. And they're composed mainly of manganese. They're also called manganese nodules, but they contain lucrative amounts of metals that are used in today's high-tech industry for lithium batteries, windmill batteries and hybrid car batteries. These metals are nickel, cobalt, and copper. And today, the increasing demand for these metals, as well as the decreasing land-based reserves, have led to an interest into large-scale extraction of polymetallic nodules through deep-sea mining. The CCFZ itself falls outside the exclusive economic zone of any nation, and so is managed by the United Nations International Seabed Authority. And this agency has to date granted 16 licenses for resource exploration to interested parties. Furthermore, they've identified 9 areas of particular environmental interest, which are the white boxes on the map here, which are situated on the periphery of the CCFC, where no mining will take place in the future. And the idea here is that these areas can act as sources of faunal replenishment to the areas that will be mined. The Belgian company Deme holds a concession license in the CCFC, and this is the prototype nodule collector that was meant to be tested in 2019. Unfortunately, the umbilical cord failed, so this test never happened and was rescheduled for September, October of this year. But due to the circumstances, it's been postponed again toward the beginning of 2021. But this is just to give you an idea of the type of machinery that will be at 4 to 5 kilometers depth attempting to extract these nodules. This is some of the characteristic fauna, well, macroscopic fauna at least, of the Clarion Clipperton Fracture Zone, exhibiting strong associations to the nodules, because they provide something that is very difficult to find in the whole CCFC, which is hard substrate. It's largely a sedimentary environment. So we get examples of anthozoans and ophiuroids and peripherans attaching to the nodules, as well as instances of octopods that are then brooding their eggs onto a stalk sponge, which itself is attached to a nodule. So the nodules have a quite vital role in this ecosystem. In 2015, we visited the CCFZ on board the Sauna to sample 4 license areas and the area of particular environmental interest 3, which had never been sampled before. And lastly, the CCFZ is characterized by an increasing particulate organic carbon flux gradient to the southeast and increasing depth to the northwest. The identification of nematode diversity in this study was achieved using a barcoding approach, which in short involves obtaining a sediment core from the area, extracting meiofauna using Ludox flotation onto a 32 micron sieve. We amplified the 18S ribosomal locus with PCR, and then all individuals in the sample are sequenced in bulk using high throughput sequencing. And then that's the genetic data is then divided into amplicon sequence variants, ASVs, following the data to algorithm. From there, we assigned taxonomy to each ASV using the RDB classifier and custom reference databases that we have. So that's some technical details. As described in the title, we wanted to look at community structure of these assemblages. By that, we mean what organisms are present in a given environment, in what numbers, and how they relate to each other. Due to the nature of high throughput sequencing though, as well as the 18S locus and nematodes, a quantitative description of the assemblages is pretty much impossible at this point. So we focused on cooccurrence patterns, so that's what organisms are present, and phylogenetic community structure, which answers how do they relate to each other. With respect to cooccurrence patterns, this was investigated by means of checkerboard pairs. And a checkerboard pair exists when 2 species don't co-occur, which I hope is visually explanatory from the slide. So if you have many checkerboard pairs or CPs, that means that your taxa do not co-occur very often. So if the assemblage is in excess of CPs, co-occurrences are lower than expected than average, and the community is said to be segregated. Conversely, if you have a deficit of CPs, the co-occurrences are higher than expected at random, and the assemblage is aggregated. With respect to phylogenetic community structure, this was investigated using these 3 metrics, which are phylogenetic diversity, mean pairwise distance, and mean nearest taxon distance. All 3 quantify branch length in one way or another, keeping in mind that branch length on a phylogenetic tree is proportional to the number of nucleotide differences between 2 sequences. So long branches means many differences and thus lower relatedness. So in the same way as before, an excess of branch length indicates lower relatedness than expected at random, and that's an over-dispersed community. And a deficit in branch length means higher relatedness than random, and that's a clustered community. So this is what we found. Overall, 1,981 nematode ASVs covering 47 genera, the richness of which followed the observed productivity gradient, which increases toward the southeast. The 3 most abundant genera were Acantholimus, Halalimus, and Desmoscolex. Notable here is also the fraction of ASVs that are unassigned at genus level, and that's the brown color in the middle panel. This ranged to about 50% on average in each sample. So for one, that tells us that the diversity we're seeing here is an underestimate of what's present, and it also highlights the deficit of reference databases that are publicly available. Another pattern that we found is that the vast majority of ASVs were unique to each area. So we have a lot of singletons and doubletons, no shared for the most part. And that's also common to morphological assessments of deep sea environments. With respect to co-occurrences, for sake of simplicity, I'm going to tell you that in these plots, you should focus on the shape of the data points rather than the absolute value at this point. So in this plot, this asterisk symbolizes aggregation. So these are co-occurrences that are higher than expected at random. So overall, we see that nematode ASVs at the phylum level and also at genus level are typically aggregated, so high co-occurrences. That's true for phylum level, so that's the first, the left-hand column, and mainly in the genera Acantholimus and Desmoscolix, whereas Halalimus ASVs were most typically not different to random assemblage. Then with respect to phylogenetic community structure, we found overall, again, the principle is the same, looking at the shapes. Squares are a clustered community, so that indicates relatedness that is higher than expected at random, and the asterisk here does not differ from a random structured community. And for overall, we found that nematodes, ASVs, for the most part exhibited clustering, so at the phylum level, that's again the far left column, as well as in the genera, in the genus Acantholimus. Whereas for Desmoscolix and Halalimus, these were, for the most part, no different than a randomly structured community. What I found interesting and important to note from this analysis is that for both co-occurrences and phylogenetic community structure, we found differences at taxonomic level. So whether the analysis was at the level of all ASVs assigned to phylum Nematoda versus all ASVs assigned to genus Halalimus. Here in this example, a combination of clustering and randomness versus exclusive randomness. But we also found differences between the two most abundant genera. So in this case, Acantholimus, again, showing a mix of clustering and randomness, whereas Desmoscolix was exclusively random. I said that with respect to co-occurrence patterns, assemblages can either be aggregated or segregated, and with respect to phylogenetic community structure, they can be clustered or over-dispersed, and each one of those has their own reasoning. So this is what we found. Overall, we found that ASVs were aggregated and clustered, which is suggestive of the following. For one, sympatric speciation, so that kind of makes sense. If speciation is occurring in a restricted area, then taxa will co-occur and they will be more closely related than expected on average. Given our data set, though, this is very difficult to prove. Secondly, co-occurrences that are higher than expected at random suggest that facilitation rather than competitive exclusion is a force structuring the community. It also suggests that the taxa have overlapping habitats rather than discrete areas that they're inhabiting. And lastly, it says that the assemblage is defined by environmental filtering of conserved traits. And I'll take a minute to explain what's meant by that. Environmental filtering describes a state in which ecologically similar and thus phylogenetically similar taxa are the ones that have the capacity to persist in a given environment. So it's the environment that selects the ecotypes, let's say, that get to persist in any given locality. The ecological context of this work revolved around understanding the drivers of deep sea biodiversity, and I do believe that the verdict is still out for this question, and most likely there's no single interpretation that's going to be universally true for all ecosystems. But from what our data suggests, we can say that high diversity in the CCFC is likely to be driven by facilitation rather than competitive exclusion. And that's in line with the stability time hypothesis that was formulated by Sanders in 1968, which attributes the coexistence of many species due to the environmental stability that has been constant in deep sea and ecosystems over time. So in essence, it's saying that the conditions have been stable for long enough for each taxa to find their own little niche and to not bother their neighbors. therefore allowing the co-occurrence of many, many species. In that way, contemporary communities are the result of past competitive interactions, but these aren't the ones that are structuring the present assemblage. Moreover, we found that the assemblages were defined by environmental filtering, and that makes them, of course, especially vulnerable to perturbations of the local conditions as they would result from mining operations. And that's the story that I wanted to tell. In short, I don't know if I did 15 minutes or if I was very quick, but I'd like to thank Jeroen and Giovanni for giving me the chance to tell it, and I'll be very happy to take your questions. Thank you.